about facilitated transport. Facilitated transport. The word facilitated meaning that it requires a protein. It can be facilitated by diffusion. I mean, it is a diffusion that requires protein to occur. Facilitated transport requires protein. Usually the molecule, the glucose, amino acid, those are larger ones. Neither molecule is lipid soluble. So usually facilitated transport does not require ATP because it is going down the concentration gradient. But sometimes what it means reversible combination is that so if it is a membrane here, if it is a carrier protein, the protein what it means reverse transport is that or the other way around, maybe. So the idea is something like this. It will carry one molecule or one ion into the cell, and at the same time you carry another ion or another molecule outside of the cell. That's what we call reversible combination. Actually, it should be glucose. It should be glucose. Oh, urea. Okay, that makes more sense. So this is what it means. When the carrier protein binds to a molecule that it wants to transport, it will induce, by the binding of the molecule, will induce a conformational changes. That means the structure of the protein will change. In this case, it will push or eject the molecule to the opposite side of the membrane, and then after the molecule leaves the protein, then the protein will go back to its original shape to get ready for binding the next one. Active transport. Active transport, they also require carrier protein. We usually call them pumps. An active transport, it is against the concentration gradient, meaning that it is going from low concentration to high concentration. Therefore, it requires energy. It requires ATP. Again, protein in active transport, they usually call them pumps. They use the energy to move the ATP to move the molecule against the concentration gradient. One example is this sodium-potassium pump. The sodium-potassium pump is especially important when we talk about nervous system, or we will learn a lot more about sodium-potassium pump. So the phosphate attaches here, it means the ATP. When ATP becomes ADP, then you have it will produces free floating the phosphate molecule. The phosphate ion will attach to the carrier protein. When the carrier protein receives this phosphate ion, then it can have the energy to move the molecule. So this is an example of the sodium-potassium pump. So inside of the cell, the sodium, they want to, normally outside of the cell you have very high sodium concentration, inside of the cell you have low sodium concentration. The opposite occurs, inside of the cell you have very high potassium ion, outside of the cell you have very low potassium ion concentration. In other words, sodium normally wants to going from outside to inside, because outside you have higher concentration of sodium than inside. So normally sodium wants to go from outside to inside. But because this carrier uses the energy from ATP, it can transport the sodium against the concentration gradient. That means transport the sodium from inside to outside. So this sodium-potassium pump will carry 3 sodium ions from inside, and then after it binds to the sodium-potassium pump, the phosphate group will attach to the pump and provide energy, and then there will be a conformational changes. The shape of the protein will change so that the free sodium ion can be released to the opposite side of the membrane. And then after that, the potassium ion from the outside can bind to it. And remember, potassium ion is higher concentration inside the cell than outside of the cell. So potassium ion is also going against the concentration gradient. After potassium two potassium ion binds to the cell, then it also induces conformational changes, and then the phosphate group release. Then the carrier protein will release or eject the potassium ion into the cell. So remember, this is the important properties of sodium-potassium pump, because sodium-potassium pump will bring 3 sodium ions from inside to outside, but this pump will bring 2 potassium ions from outside to inside. So remember, this is 3 and 2. Bulk transport. Bulk transport, briefly mentioned about this. It is basically by exocytosis and endocytosis. They are the molecule, the interest molecule that you want to transport. It is engulfed by a vesicle, and then transport to the cell membrane called, it is called membrane-assisted transport as well. Exocytosis exit from the cell, endocytosis, and including it into the cell. So remember we talked about endomembrane structure, structure. I told you it's like shipping and handling center. The Golgi apparatus is shipping and handling center. This tells us the same thing. The exocytosis is the secretion. When the vesicle coming out from the Golgi apparatus, if it passes the quality control, it can fuse with the cell membrane and secretion occur to release the final product to the outside. And the vesicle, after it fills the cell membrane, you will include it. You will integrate into the cell membrane, become part of the cell membrane. There are organs that are specialized into doing this. For example, pancreas cell. Pancreas cell, they secrete insulin or enzyme. So pancreas, when it's the cell type of pancreas that secrete insulin is the beta cell. We call it beta cell. Beta cell, or we call it an islet of Langerhans. Beta cell, they secrete insulin. And then anterior pituitary cell is release growth hormone. Anterior pituitary secrete a lot of hormone. So because of that, it is also exocytosis. So this is what the last step of exocytosis. You can see the vesicle is basically the same phospholipid bilayer. And these are the products that you want to release by secretion, and then endocytosis. The vesicle merge with the cell membrane. After it completely merged in the cell membrane, then the content of the vesicle will be released to the outside. And the vesicle becomes part of the cell membrane as well. Obviously.